Lab 20 is blood histology. So from the name, we know there will be no models associated with this lab. And in this video, we're going to cover all of the histology you need to know on a microscope slide. So to start, we're going to review the components of blood. Most of blood is going to be made of plasma, which is that fluid portion. So when we look at a blood stain or a blood smear like this, we want to keep in mind that a lot of what we see is going to be that fluid component. So plasma is about 92% water. So the background of this slide is going to contain that plasma. And because it's mostly water, it is very transparent. It's allowing a lot of light in from the microscope, and so that's why it looks really clear. Basically, we're just looking at the space between red blood cells to find that plasma. There are a couple other components, things like albumin and globulins that we aren't going to see on a slide. So just keep in mind, everything in the background here is going to be plasma. So that's the fluid component of blood. And then we also have our cellular components, which make up about 45% of total blood volume. And that's going to be the tricky part for you on lab exam 3, is identifying different types of white blood cells or leukocytes and erythrocytes. So we'll have, most of what we see will be those red blood cells, erythrocytes. So that's what we'll be dealing with on a typical slide. So these guys in pink here, these are all individual erythrocytes or red blood cells. And if we think about the shape of a red blood cell, they are biconcave. So basically, if you think about the shape of a jelly donut, the cell is going to be thicker on the edges, and then it's going to become thinner as we approach the center of that red blood cell. And that is reflected on this slide here. So we can see there's a lighter area towards the center of each of these red blood cells. That indicates that more light is shining through because of that biconcavity. So the cells are thinner in the middle, more light's going to shine through, so they look lighter here. So that's another clue you can use to identify your red blood cells. And then the other thing we can see on a typical blood smear is going to be these little specks. These are platelets. And so we've talked a little bit about these before, but basically platelets come from a progenitor cell called a megakaryocyte. This is basically just a really big, mega-sized cell, and so this is what they look like from a greater than a 40x magnification, so maybe 100x magnification, which is beyond the scope of our microscopes. But just to give you an idea of where platelets come from, they're going to originate from this precursor cell. We can see this has a lot of nuclei, so it's a multinucleate cell with lots and lots of granules. And so what's going to happen is our megakaryocyte will split off to form our platelets. Now, we will not be able to see this much detail on our slides. This is an electron microscope. So this allows us to see organelles of the platelet. These are very, very small. So the platelet is taken up mostly by these granules, these round structures here. And they do have a cytoplasm, they do have some organelles, but we're not going to spend too much time on those. They don't have a nucleus. So platelets do not have a nucleus, and mature red blood cells do not have a nucleus. So if we go back to our starting slide, to review, we've got plasma in the background, nice and transparent. Pink stuff is going to be our red blood cells. And by the way, the reason they show up as pink is because hemoglobin within the red blood cell takes up stain. So it takes up a lot of that pink stain, and that's where we get that color from. So those are going to be our red blood cells. And then lastly, we have platelets. Now that is the easy part. Next up, we have to look at our leukocytes, our white blood cells. We have two types of leukocytes. First are going to be our granular leukocytes. So these are based on physical appearance. Granular leukocytes will contain granules. They have a grainy sand-like appearance on a slide. So we'll have three types of granular leukocytes. And then we will have agranular leukocytes. So these guys are more smooth in appearance. The cytoplasm does not contain those granules. And we will have two types of agranular blood cells. So let's go ahead and take a look at our granular types. One memory cue for these is that all of the granular leukocytes contain the same ending. So we're going to start by looking at a neutrophil. If we look at the ending here, PHIL, phil, that ending will be common with all three types of granular leukocytes. So neutrophils are by far the most common white blood cell we will see. So if I give you a slide like this and I ask you to identify a white blood cell and you don't know the answer, your best choice is to guess neutrophil because these are definitely the easiest to find. We have to search really hard to find any other type of leukocyte. While neutrophils are very common, they're also very short-lived. So they really only live about 10 or 12 hours within the blood. And all of the leukocytes we talk about are going to have an immune function. These guys are no exception. Appearance-wise, they're pretty big, so they're about 1.5 times the size of a regular red blood cell. That's kind of our comparison that we use for all of the white blood cells. And they also contain multi-lobed nuclei. So we've got this new term here, multi-lobed. Sometimes you'll see multinucleate, binucleate, multi-lobed to indicate each of these little blobs or lobes is part of one nucleus. So in the case of the neutrophil, we're dealing with a 3 to 5 lobed nucleus. And they do kind of look like beads along a string. So in this case, we've got 1, 2, 3, 4, 5 lobes of one nucleus. If you look closely here, you can also see that the cytoplasm, the pink part here, has a splotchy appearance, almost grainy. That is to indicate that there are granules within the cytoplasm. So next up, we are going to take a look at a eosinophil. And I already have one pulled up here, but after we look through these, we'll also practice looking at them on a regular blood smear. So based on the ending, then we're still dealing with a granular leukocyte. In this case, our eosinophils only have 2 lobes of the nucleus. So it's going to be a bilobed nucleus. This one is really nice because it shows those granules in the cytoplasm. So we still have that speckly, kind of grainy appearance to the cell, indicating it's a granular leukocyte. These guys are a little bigger even than the neutrophils. They're about twice the size of a red blood cell, and they are involved in a very specific type of immune response. So if we have an allergic reaction to hay, or if we get hives, these eosinophils are going to have that immune response. So they are what will induce that allergic reaction. Or if you have an asthma flare-up, something like that, these cells are going to be very active in that response. And their other function is they're going to defend against bacteria and parasites. So these eosinophils can actually release toxins to kill off parasites if they are detected within our system. So pretty cool. Very hard to find on a slide. So that is less likely to appear on the lab exam, but it's a good candidate for a PowerPoint question. So if we move on, our next type is going to be basophil, and this is that last granular leukocyte. These are very rare, so I can tell you we will look for them today, but we're probably not going to find them, and I will try to find one for you guys in class if I can, but they are pretty hard to find. Basophils look really dark on a slide. They do have a bilobed nucleus, and normally it will form kind of an S-shaped nucleus, but it's really hard to tell apart from the cytoplasm because we have so many granules. So overall, you're really just looking for a very dark stained cell that's stained throughout the entire cell. And it's just taking up too much of that stain for us to tell the difference between the nucleus and the cytoplasm. These guys are also involved with inflammatory and allergic responses. In this case, the granules within the basophil actually contain histamines. So they will release those histamines during an immune response. Histamines will vasodilate blood vessels. These guys also release heparin, which is like an anticoagulant. So between those two, we're vasodilating, we're increasing blood flow to a site of infection, and we're also preventing blood clotting. So that allows really easy access to infection sites when we have these basophil secretions. And then lastly, these guys are similar in size to our eosinophils. So they're about twice the size of one of these red blood cells. So that takes care of our granular leukocytes. Now we have two types of agranular leukocytes. In this case, our ending is site. Site meaning cell. We have two types and they're going to share that ending. So cell number 1 is called a lymphocyte. These guys are also pretty common, so we will be able to find an example on our sample blood smear today. They are about the same size as a red blood cell, so they're a little smaller than the other types we have talked about so far. And appearance-wise, they do not have granules in the cytoplasm. So what we're dealing with is a clear cytoplasm, and most of the cell is taken up by this nucleus. So they do have a really large, just regular old nucleus. No weird lobe pattern here, nothing like that. And so they're pretty easy to identify. Big hint for the lab exam, make sure you know how to find these under a microscope. We do have two types. You will not be able to tell them apart under a microscope, but basically we have B cell and T cell lymphocytes. And these are named based on where they mature. So B cell lymphocytes will mature in both the liver and the spleen. T cell will mature in our thymus gland. T for T cell, T for thymus. Basically, B cells provide humoral immunity, so they're going to travel through the humoral or fluid portion of our blood to reach sites of infection. T cells are more involved with cellular immunity, so they're going to destroy more bacteria, more cellular specific threats. If, for example, we have a tumor or any sort, maybe if we reject a transplanted tissue, if our body rejects a transplantation during a surgery, T cells will become very active in that area. And then our last type we need to talk about is our second agranular leukocyte. We still have that same ending here. And this is called a monocyte. So monocytes are pretty normal looking. Again, we don't have any crazy pattern with the nucleus. Generally, the nucleus will be bean-shaped though. So sometimes it'll look like a bean, sometimes it will look like a horseshoe. No crazy pattern here. Functionally, these are activated by T cells. So there's going to be some crosstalk with the lymphocytes that we just looked at when they have an immune reaction. And they function as phagocytes. Phago means engulf or to eat. These are going to develop into macrophages when they're active during an immune response so that they can engulf pathogens and any other sort of immune threat. They're also the largest of our leukocytes, so they're about 3 times the size of a red blood cell. They're just a little bit bigger than those eosinophils. Let's try to wrap this together now by looking at a blood smear. So this is basically what you would see if you took out a blood smear during class and put it under your microscope at 4x magnification. And what we want to do is find these terms here. So this is straight from the Yale site. I recommend practicing this way because this is basically a virtual microscope. It will allow you to practice zooming in, orienting yourself, trying to figure out what tissue you're looking at, and then trying to find specific features. So for these last couple of minutes, we are going to do just that. So we want to find an erythrocyte, a neutrophil, a platelet, a eosinophil, and a lymphocyte. That does not cover every single cell we talked about, but that covers most of what we will likely find on the slide. Anything else is really rare, so that would be more of a PowerPoint candidate. Things like not our eosinophils, but our basophils, more of the monocytes, things like that are less common. Those are good candidates for PowerPoint questions. So let's go ahead, jump in. The Yale site has helpfully pointed out some markers for us already, so we're just going to zoom in and try to figure out what cell it wants us to identify. So this is what we will see at 40x magnification. So it's still pretty zoomed out. I'm not going to zoom in further because I want you to get used to the kind of this 40x view. So first things first, we have tons and tons of erythrocytes. So everything in pink here is a red blood cell. We do have plasma, so the white space in between, this would be that fluid component of blood, plasma, and it looks like we do have a couple of platelets. So these are really tiny specks here, scattered throughout. Those are going to be our platelets. The marker is on a type of leukocyte, and we know that because it's bigger, first off, and we also have that dark purple staining indicating a nucleus. Everything else on the slide, plasma, red blood cells, platelets, does not have a nucleus. So if we see this, we know we're dealing with a leukocyte. In this case, we have a multi-lobed nucleus, and I can count 4, maybe 5, so we'd have to zoom in closer, which is beyond our microscope's capability. But we can tell there are at least 4 lobes here, and that tells us this is a neutrophil. If you're still not sure after going through that process, you can click here, and it will give you that answer, as well as a little bonus information. So that is how I would recommend you practice. So, found our neutrophil. Let's keep going. All right, let's see what we're dealing with down here. All right, this is a kind of tricky one to see, but what I'm noticing is we've still got a darkly stained cytoplasm. So we're still dealing with a granular leukocyte, and it looks like we have just 2 lobes. So the dark purple stuff you see here, we've got one on the right, one on the left, connected by this little string. So I'm guessing this is a eosinophil. Let's go ahead and check our answer here. Okay, so to confirm, this is a eosinophil. It is large. It's about 2.5 times the size of those red blood cells. We're just dealing with that bilobed or 2-lobed nucleus. So those are all things that point to this being a eosinophil. Last thing we want to find is a lymphocyte, so you can probably guess what these other markers are going to be. Let's go ahead and zoom in. Okay, so things I noticed here, we're dealing with a lot of purple. So this cell basically is all nucleus. I can see just a little bit of cytoplasm on the periphery here. The rest of it is going to be taken up by that really big nucleus. So that is your main clue that this is a lymphocyte. It's a little big, so that shape convention, that size convention will not always work. In this case, this is a large lymphocyte. Other things we can notice, we'll try to find it on that other cell, but we would expect to have a clear cytoplasm here, so no granules in that lymphocyte. All right, let's check out that last cell. If we zoom in, okay, we have what looks like another lymphocyte. So again, this is mostly nucleus with that dark purple staining. We can't really see that cytoplasm, but what we would expect if we could is we'd have a clear cytoplasm, agranular, so there's none of that speckling that we would see with the other types of leukocytes. So let's go ahead and check our work. Yep, and this is a small lymphocyte. So I recommend you repeat this process. It doesn't have to be the Yale site, although they do have some other slides here, blank ones to practice with. You can do a quick Google search of any histology you want to practice with, and you'll get tons and tons of labeled or unlabeled results, anything that you should desire to study with. So I hope this helps as you prepare for Lab Exam 3.